Hello again, this is Wendy Lyons with a brief discussion of AMP1, Chapter 4, Cellular Metabolism, Section 2, Control of Metabolic Reactions, with the learning outcome of understanding a little bit more about the regulation of metabolic pathways. As we've learned from our text, metabolic pathways are the breakdown or synthesis of biochemicals. To understand this a little bit better, we need to understand how enzymes work. First, you have catabolism, where there is one larger substrate that is broken down into two smaller ones. This actually releases energy into the system. Now, you have a large substrate that attaches to the active site of the enzyme, and once the enzyme has converted the products, you have two separate products that are released, thus releasing energy into the system. The second diagram, you have anabolism, which is the exact opposite. You have two smaller substrates that are joined into one larger product at the end. This actually requires energy from your system. Now, in both instances, the enzyme is not changed during this chemical reaction. Now, I know this looks pretty scary, but just under a generation ago, anyone who is interested in studying any pre-healthcare studies were required to memorize tables such as this one. These tables were used to represent the biochemical pathways that represent energy reactions within cells, otherwise known as metabolic pathways. Luckily today, we actually have databases that are compiled using as much as we've learned today about these metabolic pathways. This is called the Human Metabolome Database. According to an article by ScienceDirect.com, metabolic pathways and cycles are reaction chains where chemical products become the substrate for the next step. All substrates are chemically transformed. Thus, you see in the diagram, substrate 1 interacts with enzyme A, becoming substrate 2. And substrate 2 interacts with enzyme B to form substrate 3, interacting with enzyme C to form a product. Now, these are enzyme-controlled reactions that form a product. There is also a negative feedback reaction in which there is a rate-limiting enzyme where the product may inhibit the actual enzyme.